Good morning, friends. In the last lecture, we have seen about the chromosome, their number, size, and shape of the chromosome. Today, we have discussed about the detailed structure of the chromosome. Okay, the chromosome is made up of chemical composition is what DNA and histone and also non-histone protein. Non-histone protein. That is the chemical composition of chromosome. Chromosome is made up of DNA, histone, and non-histone protein. Chemical composition. Okay. Today we have discussed in detail structure of chromosome. Structure of a chromosome. Structure of chromosomes. In this structure of chromosome, the chromosome is consisting of 8 parts. The chromosome is consisting of 8 parts, and that are, for example, primary constriction, also called centromere, primary constriction is also called centromere. And here second one is chromonemata, this part is called chromonemata. Then here secondary constriction, first are also called nucleolar organizer, and here this part is called nucleolus, and here satellite or the sat body. Then here secondary constriction, second, primary constriction first, secondary constriction second. And here topmost part is telomere, and here chromomere. So here a part showing chromosomes, B part showing secondary constriction and detail, details. That is the B and that is the A. Here we are also labeling A in this centromere. Here that is that parts, matrix, and here chromonemata. This is the part showing chromosome, and this is the part showing secondary constriction and details. So that in that structure of chromosome, they have consisting of 8 parts. So in that 8 part, for example, which are the primary constriction, also called centromere, first, secondary constriction, then chromonemata, chromomere, telomere, centromere, and matrix. That is the 8 parts of the chromosome. Okay, that is the 8 parts of the chromosome. Here we have discussed for the 8 parts one by one. Okay, then first part of that chromosome is the chromosome is observable during the metaphase because in this metaphase, due to the highly condensed structure of the chromosome, they have easily observed with the help of light microscope. Okay, and chromosome is easily seen in the during the cell division. Okay, here the first part of the structure of chromosome is primary constriction. Here, primary constriction. Here, first part is called primary constriction, is also called centromere. Is also called centromere. This part is called the primary constriction or centromere. In this, a typical chromosome is of two chromatids. Here, a typical chromosome is of two chromatids joined with the centromere. A typical chromosome consists of two parts and joined with the centromere. Chromatids are joined with the centromere in the primary constriction. Here, the primary constriction, or also called centromere, which is consisting of a disc-like plate, and that plate is called kinetochore. A disc-like plate which is found in the structure of chromosome. A disc-like plate is called the kinetochore. In this kinetochore, the points of the attachment of the microtubule. In this kinetochore, it is the points of attachment. Of the microtubule. We are discussed in detail in the 11th cell division. Okay, so the point of attachment in this region, like that. The point of attachment in this region of the disc shaped plate, that is called kinetochore. And here the point of attachment of this structure is microtubules. Okay, and microtubules give rise to the spindle fibers. And that spindle fibers, which are helpful to movement of chromosome during the cell division. With the help of this spindle fiber or the tractile fiber, also called the tractile fiber. Okay. So in this session, in this primary constriction, we have to study most important terms like the primary constriction, also called centromere, and that is consisting of the disc-like shape or the plate is called kinetochore. They are produced to the attachment of the kinetochore is produced to the attachment of the microtubules, and microtubules give rise to the spindle fiber for the movement of chromosome during the cell division. In this second point of the structure of chromosome, second one is also called secondary constriction. Here, some of the chromosome or the few chromosome which are also give rise to the secondary constriction. Okay, so let us see the secondary constriction. Second point, we have studied the 8 points. So here, second point is secondary constriction. Secondary constrictions. Here, we see the diagram. The secondary constriction first, also called the nucleopolar organizer, and here secondary constriction second in below the telomere region of the structure of chromosome. The secondary constriction is of two types. Secondary constriction is of two types. Okay. First one, which is of the breaking and here the breaking of the chromosome, or the also called sequent fusion of the chromosome. Okay. Breaking and subsequent fusion of the chromosome. That is the first type of the chromosome. Breaking and subsequent fusion of the chromosome, that is the first type of the secondary constriction. And second one is they are metabolically active substance. Okay. In the secondary constriction, first one, the breaking and subsequent fusion of the chromosome, and second one, they are metabolically active during the cell division. In this, if you see the structure of the chromosome, here they are give rise to nucleoli. They are give rise to nucleoli during the interphase of the cell division. That is the secondary constriction. Here, secondary constriction first and secondary constriction second. Okay, that's all about the secondary constriction. Okay, in this secondary constriction, then we have to go to the third point, and that third point we are getting sequential study in the third point. So, in this third point, the chromonemata. Third point is chromonemata. Sorry, for example, we are getting chromatids given. Chromatids study करेंगे chromatids. Okay. Here, that part is called chromatids. All parts in this part is called chromatids. Okay. The chromatids which are made up of DNA. The chromatids which are made up of DNA, RNA, and some kinds of proteins. The chromatids which are made up of DNA, RNA, and proteins. That is the chromatids. And that chromatids which are joined with the help of centromere. That chromatids which are joined with the help of centromere. For example, this is the centromere, and that is the arm or the chromatids which are joined with the help of centromere. In these chromatids which are hub into the cell division in the anaphase. Half of the chromatids which are moving toward the equatorial region to polar region. In this condition, if you see the cell structure, here they are pole region, here pole, and here equator. Okay. So the chromosomes like that, that is the spindle fiber. Okay. Here they are moving toward the like this. That is the chromatids with their centromere opposite. Okay. Like this, they are moving toward the pole region, and hub, that means only two chromatids with their centromere. Hub chromosomes. Okay. Two chromatids with their centromere in the anaphase of the cell division. So, the next point of the chromosome, and that's one 4, that is the chromatin. Chromatids, then chromonemata. This is the chromonemata. They are thread-like elongated structure. They are thread-like coiled structure, and that is called the chromonemata. Thread-like, for example, if you see chromonemata, for example, if you see the thread-like, this is the chromonemata. For example, that structure, chromonemata, that structure is called, this structure is called chromonemata. This structure is called chromonemata. Thread-like elongated coiled filamentous structure, thread-like filamentous structure, that are called the chromonemata. And that chromonemata play an important role. That chromonemata play an important role in control the size of the chromosome. Okay. That controls the size of the chromosome and bearing the various gene that is the chromata. Bearing the gene and control the shape of the chromosome, that is the function of chromonemata. Okay, that is the fourth point. Then fifth one is chromatin. Fifth one is chromatin. Here I will write that fifth one is chromatin. Chromonemata we are also seeing in this region, chromonemata. Okay, chromatin. In this chromatin, they are also like that chromatids which are okay. They are also made up of the DNA, RNA and protein. Like this the chromatin. They are also made up of DNA, RNA and protein. Okay. Then go to the sixth point. We are going to see the chromomere. Sixth point. Chromomere. See the diagram of that structure. This is the chromomere. That is the like this. In this structure of the chromosome is called chromomere, like the beaded structure, bead-like structure is called chromomere, which are parallel to the chromonemata, which are parallel to both sides of the chromonemata, and that is called the chromomere. They are parallel to the chromonemata, and this is called the chromomere. The chromomere, which are also helpful to gene bearing and gene carrier. Okay, this structure of the chromomere is also gene bearing and gene carrier. Here we will write gene carrier, which is found in the structure of chromomere. This is the helpful into the structure of chromosome and function is gene carrier. Okay. Then seventh number of the structure of chromosomes. Seventh number. What will be seventh is okay. Telomere. Seventh structure of the chromosome is telomere. The terminal region of the structure of chromosome is called telomere. This is the terminal region of the structure of chromosome. Terminal region, that is called the telomere, which along with the chromosome. Okay, that is called the telomere. In this telomere, here it allows the chromosome to get attached into the nuclear envelope other than the telomere. That is the function of telomere. Okay, it gets attached to the chromosome to the nuclear envelope. It gets attached to the chromosome in the nuclear envelope other than the telomere. That is the function of telomere. Okay. In this telomere, again one of the point. Okay, that's all. 2 points are necessary in this. So then last part of the structure of chromosome is okay. In this diagram, you will see the satellite. Sorry, we are getting firstly matrix and then study the satellite. Cell matrix. The jelly-like substances which are present into the pellicle, that is the surrounding layer of the chromosome. Here, chromosome chromatids. Surrounding layer of the chromatids is called the pellicle. In this pellicle, the jelly-like substances which are present in the structure of chromosome, the jelly-like substances which are present into the structure of chromosome, and that is called the matrix. Okay, matrix means jelly-like substances. Okay, then satellite. In this structure of the chromosome, just below the distal end of the nucleolus region, distal end of the nucleolus region, and that structure is called satellite. Okay. Today we are studying the structure of chromosome, their parts and their function in detail. We discussed the structure of chromosome. Okay, thank you.